Welcome back, guys. Today we are going to discuss this topic of biochemistry, that is our pyruvate dehydrogenase. Very simple but yet important topic of the biochemistry. So let's get started. So we are going to see first the fate of pyruvate. What is the entire fate of pyruvate that is produced in our body through glycolysis? Fate of pyruvate. So this is our pyruvate at the center. Okay. Now this pyruvate first enters our PDH complex. PDH complex. We are going to see one by one. What is this? Then converted to acetyl CoA. Okay. This pyruvate also enters through lactate dehydrogenase enzyme, and we know lactate is produced. Lactate is produced in exercising skeletal muscles. Here one NADH converted to NAD+. Very simple, we know this from glycolysis, production of lactate. Pyruvate also is acted upon by alanine aminotransferase. Alanine aminotransferase. Liver enzyme, a transaminase enzyme, and this requires vitamin B6. We will discuss about this in details in transamination in the amino acid. Now remember, this pyruvate is converted to alanine. Alanine is a important amino acid. Okay, alanine aminotransferase. This is our ALT enzyme. Okay, and finally this pyruvate also enters through pyruvate carboxylase, where in gluconeogenesis produce oxaloacetate. Here it utilizes an ATP to produce ADP with an addition of carbon dioxide and requires biotin. So we will discuss this in details in gluconeogenesis. So go check that out. So we have four fates of pyruvate, four general fates of pyruvate. Pyruvate dehydrogenase complex entering to produce acetyl CoA, lactate dehydrogenase acting to produce lactate, alanine aminotransferase to produce alanine, and pyruvate carboxylase in gluconeogenesis to produce oxaloacetate. So our topic of discussion is this pyruvate dehydrogenase complex and how acetyl CoA is produced. So let's getting into it. Pyruvate dehydrogenase. Okay. Now this site of pyruvate dehydrogenase is mitochondria. Mitochondria. Okay, where it is present. This is the outer membrane of mitochondria, and this is the inner membrane of mitochondria. Okay, here outside it is, we have our pyruvate, and this here in between the two membranes, outer and inner membrane, we have our proton symporter. This complex is exactly a proton symporter. This complex transports pyruvate inside to the mitochondria. Okay, so it is mitochondria. Now the important discussion here is pyruvate dehydrogenase is a multi-enzyme complex. Multi-enzyme complex. The pyruvate dehydrogenase is a multi-enzyme complex which consists of, remember, 3 enzymes and 5 coenzymes. 3 enzymes and 5 coenzymes. So it is a very important fact, multi-enzyme complex. So let's see what are the 3 enzymes and altogether the 5 coenzymes. Very simple discussion, nothing fancy, just watch. So there is an enzyme 1. First enzyme is enzyme 1. This is our pyruvate dehydrogenase. Okay, excuse me, I should have written here complex. Let me write a complex. So multi-enzyme complex, so it's a pyruvate dehydrogenase complex. First enzyme is our pyruvate dehydrogenase. And what are the coenzymes? Remember, thiamine pyrophosphate, active form of vitamin B1, thiamine pyrophosphate. Okay. Second is our E2. E2 are dihydrolipoyl transacetylase. Dihydrolipoyl transacetylase. What are the coenzymes? Coenzyme A and lipomide. Lipomide along with coenzyme A. So I've seen 2 enzymes and 3 coenzymes altogether. Now the final enzyme are E3. Okay, this is our dihydrolipomide dehydrogenase. Dihydrolipomide dehydrogenase. This was dihydrolipoyl transacetylase. This was dihydrolipomide dehydrogenase. Now out of 5, 2 coenzymes are remaining. First is our FAD and other one is our NAD+. Okay, pyruvate dehydrogenase multi-enzyme complex, 3 enzymes, 5 coenzymes. Very, very important. Now this pyruvate dehydrogenase complex is similar to, remember, alpha ketoglutarate dehydrogenase in TCA cycle. Okay, other one is branched chain keto dehydrogenase in branched chain amino acid metabolism. Branched chain amino acid metabolism. Branched chain keto dehydrogenase in branched chain acid metabolism. These two, as I've said, is similar to alpha ketoglutarate dehydrogenase and branched chain keto dehydrogenase. These are also multi-enzyme complexes. They also have 5 coenzymes, but the enzymes are different. Coenzymes are same, and E2 and E3 are common. E2 dihydrolipoyl transacetylase and dihydrolipomide dehydrogenase. These two are common there. Okay, these two, E2 and E3. E1 differs in all those. Remember, alpha ketoglutarate dehydrogenase in TCA cycle, branched chain keto dehydrogenase in branched chain amino acid metabolism. These are also multi-enzyme complexes. Very important. Okay, so we've seen the enzymes and coenzymes, the location in mitochondria, similarity to other enzymes. Now we'll see the reactions. It's a little bit complex, like the HMP pathway, but not that complex. So stay with me. We have our pyruvate dehydrogenase complex. So we will have our pyruvate. Okay, pyruvate being converted to carbon dioxide. It's a 3 carbon compound. Pyruvate we know. Okay, along with here thiamine pyrophosphate. This coenzyme is also being converted to, along with this step, hydroxy ethyl thiamine pyrophosphate. TPP is then converted to hydroxy ethyl thiamine pyrophosphate, and this hydroxy ethyl thiamine pyrophosphate is getting converted to TPP again back through the action of one enzyme. And what is that enzyme? See here, E2 enzyme is reconversion of hydroxyethyl TPP to TPP. And what is that? Dihydrolipoyl transacetylase. Simple. Now you are asking, where is the enzyme E1? Where does it act? It acts here. Conversion of pyruvate to carbon dioxide, pyruvate dehydrogenase, and this entire step is our oxidative decarboxylation. Okay, we have seen two enzymes here. The first one, second one in TPP2 hydroxyethyl TPP. Along with this reaction, now this reaction occurs. What? Oxidized lipomide. Okay, oxidized lipomide is getting converted to acetyl lipomide. Okay, now this acetyl lipomide is getting converted to reduced lipomide. Okay, this reduced lipomide is getting converted back to oxidized lipomide. Okay, now here we have our production of acetyl CoA. How? Now this coenzyme A enters here and converted to acetyl coenzyme A, the removal of this acetyl group, and this acetyl lipomide is converted to reduced lipomide. This is the step here where acetyl CoA is produced. And this reduced lipomide is again getting converted to oxidized lipomide. So the enzyme must act. The enzyme is finally E3 enzyme, dihydrolipomide dehydrogenase. Okay, very simple. So I've seen all the three enzymes, E1, E2 and E3, and how here acetyl coenzyme is finally produced from acetyl lipomide. Acetyl lipomide is produced from oxidized lipomide, and reduced lipomide is getting converted back. Therefore, E3 enzyme. Now, along with this step, okay, the conversion of FAD to FADH2 is occurring. Okay, FAD to FADH2 occurring, and this is also getting converted to back to FAD+. And along with this, our last step, NAD plus H plus getting converted to NADH. FAD and NAD plus were the coenzymes, they are getting used here. Okay, to FADH2 and NADH. Now the third coenzyme CoA is getting used to acetyl CoA, and the lipomide coenzyme is getting oxidized and reduced, and the acetyl lipomide intermediate is getting used here, and the last fifth coenzyme TPP is getting converted to hydroxyethyl TPP, and the three enzymes are getting in subsequent steps. It's a very interesting reaction, not at all difficult. If we say 2 to 3 times, we will remember what and where is happening. So this is the total gist of our pyruvate dehydrogenase complex and their reactions. Okay, so we'll see the summary. Summary is important here. Summary of, okay, pyruvate as one of its fate, pyruvate dehydrogenase complex produce acetyl CoA. Okay, along with this, NAD plus getting converted to NADH, which in electron transport chain translates to 2.5 ATPs. 2.5 ATPs are produced in electron transport chain with a loss of carbon dioxide, decarboxylation. So in the starting website, it's a oxidative decarboxylation. So this is the total entire summary of pyruvate dehydrogenase complex. Acetyl CoA production, NADH entry into ATC 2.5 ATPs, and decarboxylation. Important 3 points of pyruvate dehydrogenase complex. Now we'll see the regulation. All reactions have their regulations. Regulation. First is well-fed state. Well-fed state. and we know in well-fed state high insulin glucagon ratio, okay, and in this well-fed state PDH is active. First important point, PDH is active, okay, under the influence of high insulin glucagon ratio, PDH is active. So this is in a dephosphorylated state because insulin reduces the levels of kinase enzyme, so there is no much phosphorylation occurring. This is a dephosphorylated state. PDH is active in well-fed state in high glucagon ratio. Very important points. Active, dephosphorylated. Okay. Now, secondly, we have conditions. Okay, we have some conditions. First, we have increased ATP to ADP ratio. Okay, also increased acetyl CoA to CoA ratio and increased NADH to NAD plus ratio. Okay, in all these cases there is accumulation of products, and we know from basic chemistry accumulation of products drive the reaction backwards. So all this condition inhibits pyruvate dehydrogenase complex. ATP is more, inhibits, acetyl CoA produced more, it inhibits, NADH produced more, it inhibits. These are three conditions in which there is inhibition of pyruvate dehydrogenase complex. We've seen two types of regulation here, not much is there to discuss. Now we will see significances of PDH. Okay, first is this pyruvate to acetyl CoA. This is an irreversible reaction. Okay, so there is no enzyme in human body, okay, that can reverse this reaction. There is no single enzyme present in the human body that can reverse this reaction, that is formation of pyruvate back from acetyl CoA. Okay, so acetyl coenzyme A can't be converted back to pyruvate, so we come to conclusion that acetyl coenzyme A is not a substrate for gluconeogenesis. It's not a substrate for gluconeogenesis. This is the conclusion. This is the reason why acetyl coenzyme is not a substrate for gluconeogenesis. Instead, propionyl CoA enters the gluconeogenetic pathway. We have discussed in the glucogenesis video. You can go and watch that one. So this is the reason why acetyl CoA is not a substrate for gluconeogenesis, as it can be converted back to pyruvate, because there is no enzyme in human body that can reverse this reaction. This is the reason why acetyl coenzyme is not a substrate for gluconeogenesis. Okay. Then we have the excess carbohydrate. What happens when we have excess carbohydrate? Okay. Now in this case of excess carbohydrate is our glucose. Take this common example of glucose. Glucose is converted to pyruvate and 2-acetyl coenzyme through glycolysis and pyruvate dehydrogenase complex. Now this acetyl coenzyme A has two pathways, to TCA cycle or to synthesis of fatty acids, fatty acid synthesis. Okay, now this TCA cycle is very low. The chance of getting into TCA cycle is very low in this excess carbohydrate. Why? Due to depleted oxaloacetate. Due to depleted oxaloacetate, this chance is very low. So there occurs fatty acid synthesis, which combines with glycerol to form triacylglycerol. And this is stored in adipose tissue. And this is the reason why excess of carbohydrates in our body gets converted to fat. Excess consumption of rice without proper exercise or their utilization will get converted to fat. This is the reason, because oxaloacetate is getting depleted and the acetyl CO is not entering into TCA cycle. Okay. Now third significance we are going to see in chronic alcoholics. Okay, in chronic alcoholics there is less ATP production or energy depletion. Okay, now why in chronic alcoholics alcohol inhibits absorption of B1, and what is the active form of B1? Thymine pyrophosphate. This is one of the coenzymes of pyruvate dehydrogenase, the first enzyme, the most important vitamin form in pyruvate dehydrogenase complex. So alcohol inhibits the absorption of vitamin B1, that is thymine, and also in TCA cycle. Thymine pyrophosphate acts in both pyruvate dehydrogenase and also in TCA cycle. So this is a general nutritional deficiency in alcoholics. Because alcohol inhibits absorption of B1. Okay, there is less ATP production, energy depletion. So general nutritional deficiency. Okay. So now, and fourth significance we are going to see. This one is the third one. Pyruvate dehydrogenase is called a link reaction. Why link reaction we are going to see here. So this is glucose goes to pyruvate. Okay, through glycolysis. Very simple. Now this pyruvate through pyruvate dehydrogenase produce acetyl CoA. Acetyl CoA finally enters TCA cycle. Okay, so glucose enters the TCA cycle through pyruvate dehydrogenase complex through production of acetyl CoA. So it's a irreversible route between glycolysis and TCA cycle. Irreversible route between glycolysis and TCA cycle is PDH, pyruvate dehydrogenase complex. So is also called what? Link reaction. Link reaction. Pyruvate dehydrogenase complex, this reaction production of acetyl CO is link reaction. Now finally we will see some clinical significances. So most commonly the E1 enzyme. These are pyruvate dehydrogenase is most commonly affected. Okay, most commonly affected in metabolic defects. Metabolic defects, entire pyruvate dehydrogenase is affected, complex is affected. Most commonly is E1 enzyme, that is a pyruvate dehydrogenase is most commonly affected. So pyruvate will not produce acetyl coenzyme. Instead goes to lactate formation through glycolysis, lactate formation. And this we know produces lactic acidosis in case of excess glucose. Lactic formation leads to lactic acidosis. In metabolic defects, pyruvate dehydrogenase is inactive. So as a result, brain is dependent on oxidative pathways. So brain is affected. And we get neurological manifestations. Okay, this was only one clinical significance of pyruvate dehydrogenase. With this, we complete our discussion on pyruvate dehydrogenase, multi-enzyme complex, the enzymes and coenzymes. They are not so much complicated reaction. Very simple one. Its regulation, clinical significances, and metabolic significances also. Very important topic, asked every time. So please do revise them, guys. Thanks for watching. I will catch you in the next one. Good luck.